Hello, everyone, and welcome to the first session of our July 2025 All Protein Dialogue Series, a collaborative webinar initiative designed to connect and showcase all protein innovators across Asia Pacific, India, China, Australia, and New Zealand. All Protein Dialogues is co organized by GFI APAC, GFI India, and GFIC to foster regional partnerships that advance globally impactful innovation in alternative proteins. Each session brings together leaders working at the forefront of scientific research, product development, and systems level transformation. My name is Amrita Girivasan, and I'm the Scientific Engagement Coordinator at GFI India. I'm honored to be your host for today's session as we open this series focused on one of the most foundational aspects of our field, nutrition. This two-part webinar titled Understanding Nutrition in Alternative Proteins, Insights from Across Asia and Beyond will spotlight experts whose research spans across topics like nutritional formulation strategies, functional ingredient performance, and the potential of biofortification and underutilized crops to improve the nutritional profile of alternative proteins. Whether you're working on plant-based, cultivated, or fermentation-derived proteins, or interested in how all proteins contribute to healthier food systems, this session is for you. Today's session will feature expert talks from two prominent speakers. I'd like to take a moment to thank our speakers for graciously accepting our invitation to speak at this webinar. First, we'll hear from Dr. Jolieke van der Poels, Professor and Food Innovation Program Lead at Queensland University of Technology, QUT, followed by Dr. Kamal Kumar Malukani, Scientist at the Tata Institute of Genetics and Society. So after both the presentations, we'll open the floor for Q&A. Please feel free to submit your questions at any time using the Q&A box at the bottom of your Zoom screen. You can do this all throughout the webinar, and kindly make sure to mention the name of the speaker you want to direct your question to. Additionally, do keep an eye out on the Zoom chat where our team will share helpful resources and links. This session is currently being recorded. So without further ado, let's get started. I'd like to warmly introduce our first speaker. Professor Jolieke van der Poels is a nutritional epidemiologist and nutrition scientist holding a master's degree from Wageningen University in the Netherlands and a PhD from the University of Cambridge. Her research focuses on novel foods and dietary changes that improve planetary and human health, particularly precision fermentation and the increased use of pulses. Clinical trials to evaluate food safety and health outcomes are a core methodology in her research. Professor van der Poels is currently the program lead food innovation at the Centre for Agriculture and Bioeconomy at the Queensland University of Technology, Brisbane, Australia, and co-deputy program lead innovative ingredients at Australia's Commonwealth Government Food and Beverage Accelerator Trailblazer program. Professor Jolieke's talk, titled From Pulses to Precision Fermented Ingredients, Evaluating Nutrition in Sustainable Protein Sources, will talk about balancing sustainability with nutritional characteristics to optimize human and planetary health. It will illustrate and discuss nutritional attributes and health considerations for the alternative protein sector using ongoing research. I'll now hand it over to Professor Jolieke for her presentation.
Thank you very much, Amrita, and I'm just starting my presentation. Could you please confirm if that is coming through?
Yes, we can see your screen.
Yeah, thank you very much. Yeah, thank you again to the GFI team for inviting me to speak today. It's a real honor and pleasure to be here and to connect with so many people across the world. So the focus for today is nutrition and alternative proteins. I'd like to first introduce us. So we're in QUT, Brisbane, Australia. The work that I'm presenting about is embedded within our Centre for Agriculture and the Bioeconomy. You can see my colleagues here within our program. We are a very diverse group and extending discipline background areas from engineering, science, business through to health. So we're a very diverse group of researchers here in Brisbane looking into food innovation. The focus for today is nutrition, and my background is particularly as a nutrition and health researcher. So I thought I would start off by sharing some of the main paradigms of thought in the nutrition and health research world, because they're often relevant when we reflect on nutrition in a more interdisciplinary context. And so the nutrition science field really was starting off in the early 20th century when nutrients, vitamins in particular, were discovered. So it's a relatively young science, but initially the focus was very strongly on nutrients. But then as research evidence came along, it became clear that we should not just only focus on nutrients, because the foods that we eat might contain some healthy nutrients, but they might also contain unhealthy nutrients at the same time. So when we reflect on nutritional characteristics of food and what we eat, increasingly in the nutrition field, we know to focus more on the food and in fact more the dietary pattern level, where we consider the totality of all foods that a person consumes. And even beyond that, we know that it's very important that we consider food security, because not everyone has access to enough food that is healthy, nutritious, and also affordable. On the map on the top left, you can see the map of the world, where it's marked the level of severe food insecurity around the world. So there's still a lot of countries with very high levels of food insecurity, but even wealthy countries like Australia, we still have people affected by food insecurity. So it's very important that we keep these bigger picture domains in mind. And beyond that, still, we know that we need to deal with the impacts of the climate changes. We need to increasingly consider carefully the environmental impacts associated with the food that we are eating. And also, we need to consider whether the foods that we eat are fitting with the cultural types of foods, the food cultures, the preferences that people have around food, and also whether the way it's produced promote gender equity, for example. So in the nutrition field, we now know to have a lot more holistic view on what is nutrition in relation to health and well-being for people and the planet. So I'd like to illustrate some of these topics by picking out some areas of research that we're doing here at QUT, where nutrition is particularly at the forefront. And I'll start off with talking about precision fermentation, which is a novel way of producing proteins and other ingredients. So our talk today is focusing on proteins. But what is precision fermentation for food production? It's a novel way where microorganisms like bacteria and fungi get used, and the DNA that it codes for a particular protein will be embedded within these microbes, so that when you then grow these microbes in these big fermenter tanks, they will start to produce the target molecule. So that could be a target food protein that they then start to produce. So they start to produce the protein for the DNA that you've inserted into these microbial hosts. So this is a novel way of producing proteins and other nutrients that are molecularly identical to the conventional source, the conventional animal or plant source of those nutrients, but in a completely different way. So what sort of nutrients are being produced in this kind of way? So we know, of course, the main focus is on proteins, but also some companies are producing particularly lipids, also oligosaccharides, and also a whole range of other ingredients for foods that are just better or sometimes more efficient to produce in this particular way. We know that precision fermentation has the goal of reducing the environmental impact. So particularly when you think about protein sources that have high associated greenhouse gas emissions, this is going to offer a way to reduce the impacts associated with these proteins. Some people are particularly motivated by this method because it promotes animal welfare, of course. And eventually the hope is that this will just offer a cheaper and also eventually a more reliable and consistent way of producing these proteins, where conventional sources are increasingly affected by major shocks to the food system, such as climate change and conflicts around the world. So I'll declare why at QUT we're very interested in this, and that is because we have built this pipeline of lab scale development of these novel methods for precision fermentation, but supplementing that with a bioprocessing plant, which is located in Mackay, which is 1,000 kilometers north from Brisbane, where we offer the opportunity to then grow these microbes at large scale so that we can really have commercial size production research happening within QUT through to the health applications that we're offering as well. So what then is our interest in terms of the nutritional aspects of these precision fermented ingredients? First of all, of course, because it's such a novel method of producing proteins that we need to think very carefully about food safety. And we have the benefit of the FAO report that came out just only a couple of months ago, where they summarized all the main aspects that need to be considered in relation to safety of precision fermented food ingredients. The FAO report, I highly recommend it. It's freely available. I've got a main graph on the slide here that summarizes some of the main hazards that should be considered. But we also know that most of those hazards that have been identified are standard hazards, as in like they can be assessed through standard food safety laboratory methods. So in terms of research, we're particularly interested in the potential for the allergenicity of some of these proteins to be somewhat different than through the conventional production methods. So at QUT, we're embarking on some clinical work to look at immune response, antibody response, and my colleague, Dr. Jerome Ramirez, is coming at this with a bioprocess engineering point of view, and is really zooming in on the processing steps to see where there could be risk mitigation in terms of allergenicity risks. What are some other main nutritional considerations in relation to these foods? And this is such a novel, new field, so this is definitely not been researched a whole lot. But we need to, and you can see now that I use the different tiers of consideration. So at the nutrient level, we need to understand better whether those nutrients are indeed really truly equivalent to the conventional original versions. So we are planning to, we are going to do research in terms of that digestibility and bioavailability. Some of these proteins have micronutrients bound to them, like iron and calcium. So obviously they're also very important vehicles for micronutrient nutrition. So that is an important consideration at the nutrient level. At the food level, we need to keep in mind that these production methods produce ingredients, and that the total food will consist of other ingredients that will be added to the precision fermented ingredients. So we need to consider the total food and think about what is the healthiness of the total food that is being produced. Then in terms of the dietary patterns, we want to think through, well, are people actually going to buy these foods? If they do buy these foods, does that change their total diet? Does that perhaps shift their diet into certain directions? Maybe they stop buying some other foods. What does that do in terms of then their total overall dietary intake of nutrients and the diet quality? So also considerations need to be given at the dietary pattern level. And then zooming out a bit more still, that very important point of food security. Will those foods actually be available and affordable for all? That is something that is still unknown, because it's in such early stages of development, but it is eventually a very important key aspect that will need to be addressed by this field. So at QUT, we're embarking on some clinical work and preclinical research. Some of this is with commercial partners, so unfortunately I can't fully reveal all details on this. And very soon, in the next few days, we're hoping to make a major announcement. So you can follow LinkedIn if you want to if you're interested in the latest news coming through soon. But we are running clinical trials to look at some of these nutritional and bioequivalence outcomes, health effects. We're also going to use in vivo methods to look at bioavailability and again immune response. We're working with our Center for Microbiome Research because, of course, a main research question also is like, what will these foods do in relation to the gut microbiome? health effects. I'd like to now change topic a little and zoom in on some of the work that we're doing around pulses, or you might call them legumes. And legumes, in some way, are hardly an alternative protein source because, of course, they've been consumed around the world for so many years and they're so embedded in so many food cultures. So, a very well established, a good source of proteins. And we know that pulses, I'll call them, have environmental benefits. The growing process involves nitrogen fixation in the soil, so growing these pulses helps to avoid use of fertilizer, and therefore has main environmental benefits. They have good nutritional qualities, and like I said, they fit in so many food cultures. So a very good and healthy source of proteins, to the extent that a country like Australia, where pulse consumption is relatively very low, our national dietary guidelines recommend people to particularly increase the level of pulses that they are consuming. Around the world, there's a lot of research going on how can we increase people's consumption of pulses, particularly driven by the environmental gains that are being anticipated in that regard. So at QUT and in collaboration with colleagues, we are doing one particular project that is focusing on adolescents because, of course, young people still establish their food preferences and are still open to new knowledge and to new ways of consuming food. So PhD student Adeline Lanham is leading this research at QUT, and here we are particularly interested in adolescents understanding, first of all, what do they know about pulses? What do they understand about sustainable diets in the first place? We've used a national survey data to evaluate who are adolescents that consume pulses, what are their characteristics, and what are the characteristics of adolescents that will not consume pulses. But importantly, we're trying to then see how we could work at, for example, high school level environments, on how could we influence perhaps and the food environment to help increase consumption of pulses. So that's another main development, I think, in the nutrition world and health world, the realization that just knowledge itself is not often enough to change people's dietary intake, but that environments like what we call institutional environments, like schools, hospitals, aged care facilities, etc., our workplaces also are environments where people have a menu options, for example, at the school canteens, and where you can tweak what is on offer, and that is an influential way of shifting people's food preferences. So that's another way in which we consider nutritional characteristics of alternative proteins. I'd like to finish off then by highlighting some work that we're doing in relation to the environmental footprints of foods and how we adjust for nutritional characteristics in that work. I'm not sure if many people know life cycle assessment methodology. It is the core main methodology that is being used to assess the environmental footprint of anything, but including food. So it considers the full process from growing the food to transporting it to a processing facility, but perhaps a food industry, through to transportation to the customer. And it maps out in a very standardized way the environmental footprints of foods. So in terms of the recombinant proteins from precision fermentation that I discussed earlier, the anticipation is, and the current data shows, that these methods will provide important environmental gains, as I mentioned earlier, and that's been being assessed using these life cycle assessment methodologies. We do, however, need more data on actual full-scale commercial production of those proteins. But we know that a lot of plant sources of protein have generally much lower environmental footprints. But we also know that very often the criticism or the comment is, well, but the protein quality of these plant-based foods is less than that of animal sourced proteins. So in a collaborative project led by colleagues internationally out of the UK, Graham McAuliffe, we did a paper where we analysed the environmental footprints of different protein sources, but now we adjusted the impact estimates for the protein quality. So these are protein quality indicators that allow for the amino acid composition of the protein and the digestibility of the protein, thus making it a more fair comparison. You can see that the orange bars, which are the adjusted estimates, down estimate a little the impacts for the animal sourced foods and slightly up estimate for the plant-based foods. But at the same time, you can see that if you look across the whole playing field, that the plant-based protein sources still have significantly lower environmental footprints, particularly greenhouse gas emissions are much lower from the plant-based foods, even if you adjust for the protein quality characteristics. So Adeline Lanham, our PhD student, is also leading some of this work where we're now comparing a sandwich, this is an example of a type of food that adolescents would eat, whether they put ham on it or a hummus, and then she's using these LCA methods to then adjust for the nutritional value of the foods contained in that sandwich, and also even to the extent that she's considering the nutrient requirements of the target group, so the nutrient requirements of these adolescents. So you can see a graph from one of her recent analysis. She's preparing the manuscript. So these are standardized methods. You can see some of the nutrient reference factors on the slide that people have started to use to adjust environmental impacts for the nutritional characteristics. So that was very quickly using 3 domains of our research to illustrate how we are considering and interested in researching nutritional factors of alternative proteins. There's obviously a whole lot more research going on at QUT in food innovation space. Anyone interested, I would encourage you to have a look at our website and all the other work we're doing. The time is not enough to cover it all. But I'm very happy also to be in touch with people if they want more information or want to discuss anything. So I'll leave it there. Thank you.
Thank you, Professor Jolieke, for that insightful presentation on the importance of nutrition in the context of food security and diets, and also detailing how precision fermentation and plant-based sources like pulses can play a major role in health and sustainability for the global population. I request anybody who had any questions for Professor Jolieke to leave your questions in the Q&A box. Make sure to mention Professor Jolieke's name so we can direct the question to her at the very end. We will now turn to our next and final speaker for the evening, Dr. Kamal Kumar Malukani, based in Bengaluru, India, who is a scientist at the Tata Institute for Genetics and Society, also known as TIGS, with expertise in crop mutation breeding. His primary goal is to improve the nutritional value of crops, and he's currently focused on developing rice with increased protein and micronutrients. Dr. Kamal's talk, titled More Than Just Rice, Biofortification of a Staple to Combat Hidden Hunger, will cover his research in developing nutrient-rich rice varieties in his efforts to enrich this commonly consumed food, which is a staple for more than half of the population. Dr. Kamal, over to you.
Amrita, is my slides visible?
Yes, they're visible.
Sure, then I'll go ahead. So, hello everyone, I'm Dr. Kamal. Thanks for the introduction, Amrita, and thanks GFI for the invitation. So, I'm a scientist here at Tata Institute for Genetics and Society, and I will take a small chance to introduce TIGS here. So, Tata Institute for Genetics and Society, in short, TIGS, is a institute. Is it visible properly?
Yes.
Okay. So, TIGS is an institute in India which is focused on using cutting-edge science and technology to solve societal problems, particularly in healthcare and nutrition. So what we do is we do our own research, but we also work with many other collaborative partners, stakeholders to bring the solutions to the ground. TICS particularly is focused on three particular verticals: infectious diseases, rare genetic disorders, as well as crop improvement. I am working on crop improvement and I am trying to develop rice with better nutritional value. Why better nutritional value? If you look at the malnutrition in India, about half of Indian population is iron deficient, one third are considered zinc deficient, 7 out of 10 people do not get enough amount of protein in their diet, and diabetics is very prevalent in India. Unfortunately, many people do not know about it. And why we are trying to develop rice? Because rice is a staple food in India. You can see across India a very diverse form of rice being consumed. It can make sometimes up to 20% of one's plate, but sometimes it can even make up to 80% of somebody's plate. So if you develop better traits in rice, you, without any other additional intervention, can give a better nutrition to the society. If you look at this chart here, our image, so this is one of the very complex rice consumption system. This is, you can say, one of the very right side is a very simple consumption system. So the moment you go from left to right, you increase the carbohydrate in your diet. But what you lose is other macronutrients such as protein. What you severely lose is micronutrients such as vitamins and minerals. So there's a large population who actually depends on rice and some amount of some other item in their food. So if you increase nutrient in rice, that will directly add a lot of nutrients in their diet. So what are the ways to bring nutrition to the society? One is diversification of diet, which says you eat some half kg of leaves, vegetables, fruits, decent amount of cereals, grains, etc. But that is not economic for most of the population. There are other ways also, such as you give nutrition supplement, reduce the cost of legumes, or do the food fortification. They all come with their own challenges and limitations. The approach that we are using is a biofortification. So biofortification is the approach of increasing nutritional value of staple crops, about which I'm going to talk about now. So biofortification is either a genetic or agronomic practices that will help in increasing the nutrition component of the food. The simple approach is very simple agronomic practices. You add more amount of mineral in the soil, or you spray soil minerals when uh, your seed is being developed. It will have more amount of nutrients, or there are microbial applications. But these are not consistent results. Sometimes you will get decent results, sometimes you will not get. The other approach is genetic modification. You develop transgenic lines. GM rice, golden rice is one of the best example of this. But in India, they are not allowed for consumption or even production of transgenics. Genome editing is a very good tool which you can use to develop lines with better nutrition. But for genome editing, you need the background information of what are the genes, what exactly are the mutations that you need to do. And for nutritional traits, these things are not known. Then the third approach is very well tested, always use breeding approach. What do? You go ahead, screen all the available accessions across the place, see if there are any lines or natural germs that have higher nutrient value, and then you breed it in a popular varieties. The other approach is mutation breeding that we are using. So in the mutation breeding, what you do is you uh, mutagenize a large population. You take your seeds, treat with either radiation or a chemical mutagen, and develop thousands and thousands of lines. Now, among this line, there will be some lines which will have higher yield, some which have early maturation, like you can see in this case. Some will have a disease tolerance, and there might be some lines which will have better nutritional value. And those are the lines that we have to identify. For that, we have to screen the lines for this. Now, if you want to use a mutation breeding approach to develop better varieties, what is the way you do it? First of all, you want to find out what is the parent variety. It should be a commercially acceptable variety, if possible, a mega variety that every farmer will like to grow and every consumer will prefer to eat. Then you have to generate mutants. You screen for a trait of interest, get the stability of traits across multiple generations. Then if you have a stable lines, then you can go for variegated rice, and then you can try to find out what are the genes which are causing these high in nutritional traits. So in this, first of all, what we tried is we tried to find out which is the variety. So improved Samoa is a very popular rice variety. Summer Masuri is one of the very popular variety across Northeast Asia. Improved Summer Masuri is a modified version of it that is bacterial blight tolerant. Why bacterial blight tolerant? Because bacterial blight has been a very severe issue across India since many decades. Now, fortunately, this variety is also a low glycemic index variety. So it is already diabetic friendly variety. And if we add more nutritional traits with that, it will be an additional trait. Now, after that, we generate mutants. As I said in the last slide, we do the gamma radiation and generated thousands of lines. Then we have to go ahead and screen the population. The way we are using is we have developed a platform at TICS. We are calling it new TICS. This is for screening of the natural accession of our mutant population. So what we do, we get the paddy from the field. We dehusk it, get the unpolished brown rice. We polish it, get the white polished rice, and white polished rice is the form which is mostly common in India. More than 90% of Indian population preferably eat white polished rice. We go ahead and quantify it for iron and zinc using EDXRF. Then, since EDXRF is a non-destructive method, we make the powder out of same samples, use the Dumas-based approach for protein quantification, and then a part of the sample we also cook and go for in vitro digestion system, in which we treat the samples with different enzymes sequentially to see what is the sugar release rate in the sample. It's a pure biochemical approach, and at the end you will get a predictive in vitro glycemic index value. So that will tell you which are the rice varieties which have even further lower glycemic index. At the same time, we also are planning to expand the same facility to quantify vitamins as well. This we are doing in white rice. We can also do the same thing in unpolished brown rice. So ultimately, the aims that we are targeting is iron, zinc, glycemic index, total protein, or higher amount of vitamins in the rice. Now, when you set up this facility, you have to screen your population. So I'll give you an example of screening of population for zinc quantification. So when we screen for zinc quantification, first of all, we have to find out what is the content, what is the zinc content in your parent variety. In our case, it is ISM, that is improved Summer Masuri, as I said. In our trial, it showed around 32 ppm zinc because of field condition. And these traits do severely change based on field as well as environmental conditions. Now, then we started screening our mutants. And for each mutant, we screen 3 to 4 individual plants and get the average of this. Mutant 1 has some value, 2 have some other. In the same way, you screen thousands of mutants. I'll show you the result of one of such trial in which we screen around 1500 lines and find out what is the distribution of these lines. So y-axis shows you what is the total number of lines that shows that particular value, and x-axis shows you what is the zinc concentration. So most of the lines are around 28 to 32 ppm zinc. But there are some lines which have up to 40 ppm zinc as well. So these kind of lines are the one which are our candidate high zinc lines. Then we screen the same population for iron as well. And we have some lines which are slightly high iron content. We screen the same line for protein content. And we have candidate lines which have more than 12% protein content, while in the parent, it is generally below 9-10%. So these are the ways we screen our different traits. What do you do when you have candidate lines? That is the one I'll focus it now. So as I said, we screen the traits. Then we have to check if the trait is really stable across multiple generations or not. Then if it is really, really stable, then you can go for varietal release. You can go and try for gene identification and then use different approaches to bring the trait in other varieties. I'll just quickly give you a gist of how it is done. So as I said, we screen the population for nutritional traits. This is one of our old result where we screen a mutant population and the parent has about 19-20 ppm zinc in that season. Mutant have up to 27-28 ppm zinc. You go ahead, take the best plant seeds out of this mutant and cultivate it for next season. You get lines again which have better zinc. You will have many lines which will not show you better zinc than the parent. So you discard those kind of outliers every single season and continue to grow it for next few seasons. Ultimately, you will have a few lines, just a handful of them, which will give you consistently high nutrition content than the parent across multiple generations, which we have in this case. Now, what do you do when you have stable lines like this, which are showing you higher nutrient after that? You can go for national trials. So in India, we have a ICR based national trials in which you submit your seeds for trials. They will screen it across more than 30 locations and see if your variety is showing better zinc or better nutrients than uh, checks as well as controls. And if it is the case, then they will go for varietal release. So that's a very standard procedure. They have to do it across 30 locations for more than 3 years. And if and only if it passes through it, it would go for varietal release. Then this is for the variety development. As I said, we can also try to find out what is the gene causing this. To find out what is the gene causing this, first of all, you find out what is your high zinc mutant, the stable lines. You find a very diverse kind of variety which has low zinc content. You cross both of them, develop F1. I don't want to show you results here, but because we are not published yet, but you develop F1 here. Now you sell this F1 and you get this kind of Mendelian segregation where you have hundreds and hundreds or sometimes even thousands of F2 lines. Now you screen all of them for your zinc content. You will have a few which will have very low zinc. There will be few which will have very high zinc. Then you select top 10 or top 20 out of those. Use the NGS to sequence the bulk of those. Compare the sequences of your top 20 and bottom 20. And then you will have this kind of a Manhattan plot where you will see y-axis will show you log p value of association of a mutation with your trait of interest. Now, in this one, you can select what is the 95% or 99% or 90% confidence interval, and based on that, you can select which are the lines which are really your candidate mutations that are there. Then what do you do when you have the candidate mutations? You develop markers to see if the markers or the genetic loci mutations are going ahead with your trait of interest or not. You also go back and check if those mutations are really present in your mutants or not. And if those traits are present, you will end up with certain loci which are contributing to trait of interest. What is the advantage of that? First of all, it will help you find out the loci where the mutation is. And if you find out that, you can easily use the breeding to bring the trait in a different varieties. The second advantage is if you really know this exact mutation is the cause of mutation, you can use genome editing to even in a faster way develop the same trait in a different types of variety where you can actually stack multiple traits in a very fast way. So as a summary, what I can say is we have developed low GI as well as high zinc content rice, which we hope soon will go for national trials. We have candidate low GI as well as high protein lines and partially high zinc lines, which hopefully will be ready for national trials in next few seasons. One thing I'm not showing here is we have tested what is the effect of polishing and washing on rice, because as I mentioned previously, more than 90% of Indian population uses white polished rice for consumption. So when you do the polishing, what we have observed is there is very severe loss of iron and there is a significant loss of zinc as well. In most of the cases, we lose more than 80% of the iron the moment we polish it. Similarly, the very common practice in India is you cook the rice in an excess amount of water or soak it for many hours before cooking it. In both the cases also, we really lose a lot of iron. So one more thing is there's a chance that these practices can reduce iron nutrients. Those are the aspects we are looking at it right now, and soon hopefully we'll publish this part. Then one thing which is very clear is if you diversify your diet and try to add more different types of food items, that for most of the cases will help you get enough amount of nutrients from our food. So what we want to actually do in the future, we are trying to develop rice with better minerals, better protein, but down the line we also want to develop rice with better vitamin value. Develop ultra low GI. We have already a low GI rice variety. We want to develop an ultra low GI, which will be even beneficial for diabetic patients. And then we want to stack all these traits in a single variety, which will have better protein, zinc, iron, and vitamins as well, plus hopefully ultra low GI. Then there's another aspect where we have various neglected crops. Like these are the crops grown in certain regions, certain localities, and we are trying to always find out the local NGOs or institutions who are trying to conserve those kind of crops. And then we want to screen those for nutritional value and try to bring them in a national level if those are really very good nutritional benefits. And then one thing I did not mention here is we are trying to reduce anti-nutrients in unpolished rice, and down the line we'll also consider the same for millets as well. With this, this is my short team, and I'd like to thank all our funders and collaborators. We have collaboration along with CSI, CCMB, Institute of Rice Research, as well as National Institute of Seed Science and Technology, and Good Food Institute for this opportunity. If you want to reach out, please scan this QR code and you'll get my email ID and other details. And thank you all, and we will be very happy to answer your questions.
Thank you, Dr. Kamal, for that insightful talk. It was really interesting to learn about how rice, which is also a less explored plant-based protein source, would be enriched nutritionally to serve as a more functional and versatile ingredient, especially considering how much of a staple it is across Asian countries. Okay, so we can now move into our Q&A segment. Please continue submitting your questions in the Q&A box with the name of the speaker that you want to direct the question to. So we actually have a few questions right now in the Q&A box. And the first one I think is open to both the speakers. So it's from Michael, who's joining from Uganda and is part of a local community organization that works on climate action and sustainability initiatives. And Michael would like to know, how can the nutritional insights from alt proteins in Asia and beyond be applied to the African context, particularly in Uganda? And what role can all proteins play in addressing malnutrition and food insecurity in Uganda? So this is open to both our speakers.
I'm happy to comment as a first start. Thank you, Michael. They're important questions, and I think at a most basic level, when we think about alternative proteins and their nutritional value, a lot of these nutritional characteristics are generic in the sense that they will apply to people globally. But saying that, and linking to what I said earlier, that as nutrition and health professionals, we need to consider the totality of people's diets. And we also know that some nutrients in a food can interact positively or negatively with other foods and nutrients that are also in the person's diet. Some nutrients get inhibited for their absorption, some nutrients get promoted for their absorption. And then obviously the total diet of an average person in Asia will be different from an average person in Uganda, etc. So we need to always have evidence that is context specific. And yes, therefore, we also will need to do this research in Uganda as well. At the same time, I think some of what we're doing will have generic application as well.
I'd like to add on that exactly as Jolieke said. What work we are doing is a lot applies not just to our Indian or say local context. There's knowledge coming out of it which can be applicable at a global scale. So it's not just one country or something. The results that we will be coming out will be useful for all the countries, that maybe you can use the similar approaches to enrich your diet with better nutrition, or modify your local cost for better nutrition.
Awesome. Thank you so much to our speakers for answering that question. We have another one here for Professor Jolieke. So this attendee wants to know if you could recommend any new or upcoming movements in next-gen protein and precision fermentation and novel nutrient areas that you see so far. So any potential areas that could be explored.
My immediate response hearing that question is I'm very excited by these initiatives to find alternatives for example, palm oil, where we know that palm oil is such a cause of deforestation in the world, and poor working conditions, poor wages, etc. So there's big concerns about conventional palm oil production. So we now have precision fermentation startup companies, companies trying to produce palm oil or equivalent products, and that would be a major gain, right? So those initiatives where people are really trying to tackle currently, problematic food sources, that excites me. And there's so much development in this field. It's hard to keep track of what everyone is doing. I mean, it develops all the time. So there's a lot of exciting work. You can't answer that in just 30 seconds, but that is the first one that came to mind when I read that question. Thank you.
Thank you. There's one more question for Dr. Kamal, and then another question for Professor Jolieke. So, Kiao from Myanmar asks to Dr. Kamal, among three different biofortification methods, how feasible are agronomic practices for developing countries for smallholder farmers to adopt the practices while also adopting climate smart agriculture practices?
Very nice question. Yeah, thanks, Q, for the question. So that actually is the very easiest approach to do, but the issue with that is uh, it results vary from location to location and environment to environment. So you really do not get consistent results across many places. Additionally, the yield is very low. So if you really spray a huge amount of minerals, then you end up with some 1 to 5% of those in the game. So you will get a slight higher amount of minerals, but the yield in terms of efficiency you get out of it will be very low. So that one has to optimize at the local scale with particular varieties, because these practices do have effect of environment, soil nutrient content, as well as variety. So these things you have to keep in mind when you are testing this.
Perfect. Thank you so much, Dr. Kamal. Our next question is for Professor Jolieke. As your research spans both plant-based and precision fermentation-derived proteins, what are some key challenges in comparing their nutritional value on a common scale?
Yeah, that's a very interesting question too. They're so vastly different, right? On one hand, you have this completely novel method that produces ingredients, so not final foods, but ingredients that will go into an overall final food. And we have some examples, like in the US. We didn't talk much about regulation, not at all yet, but obviously the regulatory environments are a big determinant here at the moment in terms of where these products come to market. But in the US, we do have some of these products on the market, and so we need to understand better what the final products will look like that are going to be produced. But then still the precision fermented ingredient is one of the ingredients. So it'll be really hard to make generic statements because there'll be so many different types of products that will food products that will be produced using precision fermented ingredients. And on the other hand, then you've got the traditional, pulses, like a crop, like a food source that has been around for thousands or millions of years, probably, that is just available everywhere. There's no technology, you just need to be able to grow it, and it's accessible. And perhaps linking back to the first question from Uganda, what I also wanted to comment on is, what I said in terms of food security, that is a real challenge in terms of the precision fermented methodologies. At the moment, we have all these startup companies or some bigger companies are buying into it. We even have traditional dairy companies buying into it because they see a future in it. But it is within company ownerships at the moment, and so the availability is locked up sort of at the moment, and that will really need to be addressed. And that's where it's so vastly different from something like, pulses legumes that are available around the world. So it's challenging and so different, but we need to consider it all, right? Because there's never one solution.
I think that captured the answer to that really, really well. And I have one more question here for uh, both speakers, actually. Um, they would like to know if there's any guidance or resources on how mushrooms are being explored for protein R&D, either for nutrition or other applications. Just before our speakers chime in, I would also like to say that um, to dear Biplab, we have a bunch of resources on our website as well that you could take a look at. All of those are open access, and I'm sure they would offer multiple deep dives in addition to the webinars that we have. But I'll pause here and let our speakers chime in as well.
Dr. Jolieke, you would like to add first? You're mute.
Sorry, you can go first, Dr. Kamal. Thank you.
Yeah, thank you. So, Biplav, it's a very good question, and we are really considering that thing because it's a very early stage. I don't want to reveal that thing, but we are planning to develop mushroom which can actually be used more locally by the farmers using different types of waste products. And that kind of thing, if we can streamline the industry from mushroom cultivation to the protein extraction, while removing the hurdles that a farmer face for cultivation those is we are really having a lot of brainstorming session. Please feel free to reach out to me if you want to discuss it further.
Yeah, I will give a very similar answer. That is, we definitely within our group of food innovation within our CAB Center have an interest, and we have ongoing research mushrooms, and a big theme in our group is also using side streams, waste streams from current production processes to value add, and that's where also mushrooms are being looked at in terms of substrates for mushroom growing. We haven't taken that quite to the clinical stage yet, but it's a very growing area of research. One of my other PhD students is looking at in a clinical setting around muscle function, and we know that mycoproteins are really coming through as being beneficial for muscle function and muscle health. So very much a very interesting and important field of research. Thanks.
Thank you so much. And I think we can take one more question that's here in the Q&A box for Dr. Kamal. How do you balance improving nutritional content with maintaining yield, taste, or other agronomic traits that farmers care about? And I'd also like to add a small part to this question. If you've had any interactions with farmers before on related to your project on biofortification of rice varieties, we'd also love to know if there have been any specific reactions from them on your project, and if there's any expectations that you have on how they would react to this project.
So first I'll answer this question, then I'll answer your part. So one of the key cross-check in between developing the varieties, since we grow it across multiple generations, is testing for other traits as well. All the agronomic traits, which include yield, flowering time, all of this. We do make sure the mutants that we are selecting to go ahead do have better agronomic traits. So the traits that are really present in the parent variety. So that we always cross-check. Then, when we have lines which are ready to go for national trials, we have do a third-party assessment where they are institute, where you can send your samples, and they will do all the taste, cooking quality, and aromatic taste. All of these testing they do it, and they give you the report and compare it with the best varieties that are available in the market, which are famous varieties. And then you can see if your line is really fitting up in these criteria. Then only you should go for national trials. Now, coming back to your part, yes, we do have some farmers interaction every now and then, and for all of our researchers across districts, we always involve stakeholders on board before even starting the program. So stakeholders like NGOs, farmers group, particularly breeders in our case, who directly talk to farmers or farmers group. So we take them on board, and we have collaborations with many such bodies, and we take their inputs to do it. What do we do and what is the need? And based on that, what could be the way to address it? Now, farmer's reaction is as long as I am getting a good yield, and hopefully if I'm getting slightly better price, I'll prefer this. Okay, so that is one thing that even Government of India is trying to see, if farmers who are using biofortified varieties, if they can get slightly better benefit for the crop, that they will, and then they will prefer to grow food with better nutrition.
Wonderful. Thank you. I think that answered the questions that we had. And before we close off, there's just one more question from Aaron from GFI APAC for Professor Jolieke. Regarding the work your team did on protein quality assessments corrected for environmental impact, have you considered blended meat products where animal products are blended with plant-based products?
The short answer is no, but that would be really good to do. Yes.
Amazing. Okay, I think with that we've answered our questions, but I'd like to remind our audience that Professor Jolieke and Dr. Kamal can be contacted via their email IDs if you have any other questions as well. And with that, that brings us to the end of part 1 of the All Protein Dialogues webinar, Understanding Nutrition in Alternative Proteins, Insights from Across Asia and Beyond. A huge thank you to our speakers for sharing their expertise, and to all of you for joining us today. We hope this session was insightful and sparked some enthusiasm in you in understanding nutrition, specifically in the all protein context. Please do be sure to join us on July 16th, which is next Wednesday, for part 2, where we will continue this discussion with new expert speakers exploring the same topic of nutrition in all proteins. You'll also receive a follow-up email with the recording of today's sessions and the key resources and information on how to register for part 2. Thank you once again to our speakers, attendees, and the GFI India, GFI APAC, and GFIC teams for the support. And we look forward to seeing you next week. Thank you so much.
Thank you. Bye-bye.